Good afternoon, dear students. Today we are going to discuss about toxoids. In the last lectures, we discussed about conventional vaccines, that is how production of BCG vaccine is done and how production of polio vaccine is done. Today we are going to discuss about toxoids and especially toxoid, that is tetanus toxoid and diphtheria toxoid, and how to produce anti-tetanus serum. Now let us understand what a toxoid is. Now, exotoxins are proteins. Okay. Now, there are certain organisms which, when enter inside the body of the host, liberate their toxins called as exotoxins, and the exotoxins are responsible for causing some type of diseases like like tetanus, or you can say diphtheria, right? Now, what are these exotoxins? They are the toxins secreted by the organisms responsible for causing the disease or infection. Now, exotoxins are proteins which lose their toxicity when they are treated with various chemicals like formaldehyde or formalin or phenol, etc. Okay. So now, when we want to produce a toxoid, okay, why do we need a toxoid? For prevention. Why do we need a toxoid? For prevention of the disease, right? Now, how the toxoid is prepared, that we are going to discuss here. Now, exotoxin proteins are having toxicity as well as antigenicity also. Now, exotoxin proteins lose their toxicity when they are treated with chemicals like formaldehyde or formalin or phenol. Or they can be modified chemically so that they do lose their toxicity. Their toxicity is destroyed, but they retain their antigenicity. That means they can behave as antigens. Such exotoxins whose toxicity is destroyed but antigenicity is retained are known as toxoids. Okay. Toxoids have the ability to stimulate the production of antibody, which neutralizes the toxicity of the toxin when administered into the body of the susceptible host. Okay. So toxoids can stimulate antibody production, right? And the antibodies that are produced against the toxoid can neutralize the toxicity of the toxin. This important property protects the susceptible host from toxigenicity. Two types of toxins are commonly toxoided. They are diphtheria toxoid and tetanus toxoid. The first one that we are going to study is production of tetanus toxoid and then production of anti-tetanus serum. So let us first discuss how tetanus toxoid TT is produced. Now, the general characteristics or etiology of tetanus. Tetanus is caused by Clostridium tetani. So Clostridium tetani, the bacteria named as Clostridium tetani, is the causative agent of tetanus in human beings. Von Behring and Kita Sato in the year 1889 first synthesized tetanus toxin successfully. Okay, they synthesized the toxin successfully. Clostridium tetani is widely distributed in soil and residues in the intestine of man and animal, in the intestine of animals especially. Okay, so because it's a soil bacteria, it is gram-positive, anaerobic. The most important thing is it is anaerobic spore-forming bacilli. It is non-capsulated, motile with peritrichous flagella. The bacteria is sporulated. Okay, it is anaerobic sporulating bacteria. So spore forming, spores are terminal in position, giving it a typical drumstick-like appearance because spores are terminal and bulging. Okay, the medium which is used to cultivate Clostridium tetani is RCMM, Robertson's cooked meat medium, blood agar. On blood agar, it will produce hemolysis. Egg yolk medium. That means nutritionally rich media are used for cultivating the bacteria. It is indole positive. It doesn't ferment sugars. It liquefies gelatin slowly. The spores of the bacilli are heat resistant, as well as chemical resistant, desiccation resistant, and so on. So actually, it's a very resistant kind of bacteria because the spores are resistant. And the spores are killed only by autoclaving at 121 degrees Celsius for 15 minutes. Now let us study the production of anti-tetanus serum. Okay, first thing is preparation of inoculum. So we had to grow the Clostridium tetani. So Clostridium tetani is anaerobic spore-forming gram-positive bacilli. It is maintained in the culture collection center in the form of spores. The soil is the most common source of Clostridium spores. The selective strains, which give highest amount of exotoxin production, are maintained in stock culture, and they are used for the preparation of inoculum. The first step in the preparation of inoculum is to incubate the spores from the stock culture into a favorable media. The spores are allowed to undergo germination into a favorable media. Generally, we use blood agar. It is known as germination media because the spores germinate in blood agar. Once the spores germinate, then they are transferred to RCMM, Robertson's cooked meat medium. The inoculum thus prepared is then utilized for the production of exotoxin. So the first step is inoculum development. That is, you take the spores, which gives higher exotoxin production from the culture collection center, allow the spores to germinate by using a germination medium like blood agar, and then transfer it to Robertson's cooked meat media so that you get a sufficient amount of culture of Clostridium tetani. Right. The next step is production of exotoxin. The fermenter that are used for exotoxin production is made up of either aluminium or glass. Iron we do not use because iron is toxic, and that's why we don't use stainless steel fermenters. The fermenter is sterilized before production of exotoxin. Specially designed synthetic medium is used for the production of Clostridium exotoxin. The medium contains maltose, potassium dihydrogen phosphate, dipotassium hydrogen phosphate, magnesium sulfate, and aluminium sulfate. In addition, Clostridium tetani also requires various amino acids for its growth, like lysine, arginine, leucine, isoleucine. Vitamins like riboflavin, thymine are also added. After sterilization, the medium is inoculated with 20% inoculum from the RCMM medium. The organism is anaerobic, and hence we have to maintain strict anaerobic conditions. Agitation is allowed in the beginning for uniform mixing of media and organisms. And then we have to maintain the pH in the range of 7.2 to 7.8. Optimum temperature is, of course, 37 degrees Celsius. Fermentation duration is 4 to 7 days. Actual production of exotoxin starts within 24 hours itself. Now, the next step is, once the organism starts growing and starts producing exotoxin, the exotoxin gets accumulated in the medium. Now, the next thing is we have to convert the exotoxin. Is very toxic. It is very potent. Okay, it can cause toxicity in human beings. So the exotoxin needs to be toxoided. Toxicity from the exotoxin is removed and antigenicity is retained. The process is called as toxoidation, conversion of exotoxin into toxoid. Toxoidation is a process of conversion of exotoxin into toxoid. Fermented media is harvested for recovery of exotoxin. You collect the fermentation media and harvest the exotoxin. Toxin is secreted by the cells and excreted outside, and hence it is present in the liquid phase in the medium. The bacterial cells are separated from the medium by centrifugation or by filter press. Separate the bacterium. Actually, the exotoxin is present in the liquid medium, liquid phase. So separate the bacterial cells by centrifugation or filter press. The fermented broth is recovered. Okay, so you pass it through centrifugation or filter press, cells will be removed, recover the fermented broth, and then it is subjected to purification. Generally, absorption method is used for separation of toxin from fermented broth. Exotoxin is highly potent and hence cannot be used for the production of toxoid. Exotoxin being very highly potent cannot be used directly to be given to patients, right? That is why there is need of toxoidation, removing the toxicity but retaining the antigenicity. Exotoxin is having hapto-4 group and toxo-4 group. Hapto-4 is responsible for antigenicity and toxo-4 for toxicity. We need to remove the toxo-4 group. Hence, the exotoxin is treated with formalin or other such chemicals to destroy its toxicity. The detoxified product obtained is known as toxoid. It is also known as modified exotoxin or formal toxoid because toxoidation is done by you adding formalin. So it is also known as formal toxoid. Okay. So once you destroy the toxicity of the exotoxin, whatever preparation you have is called as toxoid. The toxoid is then precipitated out by adding ammonium sulfate. It is then centrifuged and concentrated by ultrafiltration. Okay, you precipitate it by ammonium sulfate, centrifuge and concentrate by ultrafiltration. It is then dissolved in a particular sterile liquid to required concentration, dissolved and diluted to required concentration. And then the purity and potency testing is done, and then finally ampule filling is done. Once the purity and potency testing have been passed, then ampule filling is done. The ampules are sealed and labeled. How to purity and potency testing is done? The diluted toxoid is now tested for purity and potency. Purity is detected by injecting the toxoid into susceptible animal. If the animal shows any symptoms or dies, it means detoxification is inadequate. Under such situation, the entire toxoidation process is repeated. If the animal does not develop any symptom, that means the toxoid is ready for vaccination purpose. Potency is calculating the required amount of dose of toxoid that is to be administered into human beings so as to generate antitoxin production, so as to stimulate antitoxin production. So potency is to be determined. After the satisfactory purity and potency testing has been done, the toxoid is filled in ampules. It is sealed, lyophilized, and labeled. The label should indicate the date of manufacturing, the date of expiry, the storage condition, the amount of toxoid present, the name of the toxoid, the abbreviation, etc. Okay, this is how toxoid is prepared. Now, once tetanus toxoid is ready, we go for anti-tetanus serum preparation. How? The commercial production of antitoxin, that is anti-tetanus serum or anti-serum, is carried out in healthy horse. The healthy horse is immunized with either the series of injections of appropriate known concentration of toxin or toxoid. So commercially, anti-tetanus serum is prepared by using healthy animals like healthy horse. Horses are immunized by injecting a known concentration of toxin or toxoid. Initially, a small dose of toxin or toxoid is injected, and slowly the dose is increased at each of the several successive injections. And then after 7 days, blood is withdrawn from the animal by trial bleed, and if the trial bleed shows a satisfactory amount of antitoxin in blood, then horse is bled from external jugular vein. First, after 7 days, small amount of blood sample is collected and testing is done. If antitoxin is sufficient amount, antitoxin sufficient is present in the blood, then the horse is bled from external jugular vein. Then a lot of blood is collected. The blood is separated into two fractions, RBCs and plasma. Plasma is partially digested with pepsin to remove the cell debris, other impurities from this fraction. And what we get is antitoxin. Antitoxin is precipitated out by digestion of plasma with ammonium sulfate. Okay. So the plasma is subjected to digestion and antitoxin is precipitated out. The digested plasma is centrifuged, dialyzed in water, concentrated by ultrafiltration, and ultimately is brought to desired concentration level. And after control testing, and after acquiring satisfactory result, antitoxins with different potency level is filled in ampules. Ampules are lyophilized, labeled, packed, and sent to market. One problem with anti-sera or plasma, this is antitoxin, is delayed hypersensitivity. Some patients may develop delayed hypersensitivity reaction, severe allergic reactions, since antitoxin is protein in nature. It occurs in patients who are immunodeficient, okay, or those patients suffering from immunodeficiency diseases, and also sometimes in normal individuals, because anti-serum is prepared from horse, okay. Anti-tetanus serum gives artificial passive immunity. Okay, ATS gives artificial passive immunity. It contains ready-made antibodies, ready-made antitoxin. It is an antitoxin which neutralizes tetanus exotoxin in multiple proportion. It immediately supplies antitoxin required for neutralization of toxin, and hence it is used only in case of emergencies. If you want to immunize a person against tetanus, we give tetanus toxoid is injected into the body. But if a patient is in advanced stage of tetanus disease, and there is an emergency to treat the patient, in that case only anti-tetanus serum is used, because hypersensitivity reaction may develop. It doesn't always develop, it may develop. Okay, so anti-tetanus serum or antitoxins are directly used, or they are ready-made antibodies, ready-made antitoxins required for neutralization of toxin, and hence used only in emergencies when the patient is in advanced stage of the disease. It is most useful in immunodeficient patients. Okay, so let us see the flow chart. So the spores of Clostridium tetani, they are germinated on blood agar, then they are transferred to Robertson's cooked meat medium, and then they are incubated at 37 degrees for so and so. Okay, after germination, transferred to for exotoxin production. During exotoxin production, they are incubated, temperature is maintained, pH is maintained, and exotoxin is ready in the liquid broth. Then from the liquid broth, the exotoxin is separated, the cells are separated, and then the exotoxin is subjected to toxoidation by given formalin treatment. This toxoid or exotoxin is injected into horse. Here we are studying anti-tetanus serum production flowchart. The horses are injected with toxin or toxoid, go on increasing the dose and all. Then trial bleed is done. If the trial bleed contains sufficient amount of antitoxin, then the horse is bled from external jugular vein. The blood collected is separated into two fractions, RBCs and plasma. Plasma is first digested in pepsin, treated with ammonium sulfate, precipitated out by treating with ammonium sulfate, centrifuged, dialyzed in water, subjected to ultrafiltration, and after control purity potency testing, ampule filling is done. Then the ampules are lyophilized, labeled, packed, and discharged to market. Okay, this is how anti-tetanus serum is prepared. This is all about anti-tetanus serum production and tetanus toxoid production.